AS Level Biology 9700, Paper 1, Variant 3, October, November 2024, question number 11 to question number 20. Students, this is the second video on this paper. In the first video, I had discussed the question number 1 to question number 10. Now, question number 11 to 20. So, I'm moving towards the question number 11. And question number 11 is the student investigated the effect of substrate concentration on the rate of an enzyme catalyzed reaction. The student plotted the results in graph. So you can see on one side, on x-axis, they have given us the substrate concentration. On the other hand, on y-axis, they have given us the reaction rate per molecular DM in seconds. So here I would like to discuss first of all, the question is what is the Km for this enzyme catalyzed reaction? So first of all, what is Km? Km is the Michaelis-Menten constant or Michaelis constant, which is defined as the substrate concentration. Substrate concentration at which substrate concentration at which the reaction rate is half of the maximum reaction rate So here you can see that on screen, that is the reaction rate increases with substrate concentration and then levels off. So it is levels off over here. It's level off over here. So here you can see this is 30. The reaction rate is 30. So we have to take its half. Its half is 15. Its half is 15. You can see. This is 15. And half of 30 is 15. So it is 12. So the substrate concentration, we have to find out the substrate concentration. So the substrate concentration is 12 at half of the reaction rate. So half of the reaction is 15. You can say the reaction rate is levels of. This is the level of at 30. So this is 30. So half of 30 is 15 and 15, the substrate concentration is 12. So it means the answer is A. So the answer is A, B, C, and D are not the correct options. We have calculated it from the graph. Now the next question is question number 12, and question number 12 is, which statement could be used to describe molecules and antibody molecules? So first of all, you know the antibodies and enzymes are the protein in nature. So they have given us the three statements, and these three statements are, the first statement is hydrogen bond stabilizes the structure of the protein and are important for it to function efficiently. So enzyme molecules and antibody molecules, we have to describe with the help of these three statements. So the statement first, uh, according to the enzymes, first of all, enzymes, they are protein in nature and their 3D structure is crucial for their catalytic activity. So hydrogen bonds play a significant role in stabilizing the secondary and tertiary structures of the enzymes. That is the in primary structure, there are straight chains. In secondary structure, alpha helicals and beta plated sheets. And the third type of structure in which the 3D structure is tertiary structure is involved, the 3D structure. So in this case, the hydrogen bonding is very important. So it is, you can say this hydrogen bonding is crucial for the catalytic activity of the enzymes. And on the other hand, when we talk about the antibodies, so antibodies are also proteins, immunoglobulins. So they have a specific 3D structure. So that allow them to bind to antigens. So hydrogen bonds in both cases are vital for maintaining their structural integrity and the ability to recognize and bind to specific targets. So I would like to say this is the correct option. Correct statement. Statement number 1 is the correct statement. And second statement is hydrophilic R group point to the center of the molecules and cause it to curl into a spherical shape. Hydrophilic means water loving. So water loving will not point in the center of the molecule. So they will not curl up in the spherical shape. So these, according to this second, I would like to say, because it is incorrect. Why it is incorrect? I already told you. That is the R groups. If they are hydrophilic in nature, they will point outside, not in the center. They will not in the center. They will tend to be on the surface of the molecule. So when they are on the surface of the molecule, so they will not cause it to curl into spherical shape. So this statement is not correct. Then we move towards the statement number 3. The tertiary structure of the protein molecule plays an important role in the functioning of the protein. Already told you the tertiary structure of the protein give a 3D structure to the protein molecules, whether they are enzymes or other types of proteins. So tertiary structure play a very important role in this case. So I would like to say this statement is true for both enzymes and antibodies. So according to these statements, you can see the C is the correct answer. B, A, and D is not correct options. Only C is the correct option because the statement number 1 and statement number 3 is correct statement. So they according to these statements, the answer is C. Now I would like to move towards the question number 13. Which description identifies a reversible non-competitive enzyme inhibitor? So here I would like to say that there are 4 options. It can attach to the active site. If the you can say enzyme reversible non-competitive will not attach to the active site. It will not compete for the active site. So the competitive enzyme will always try to attach on the active site. So according to this, we have to discuss about non-competitive. So A is not the correct option because this is the property of the competitive enzymes inhibitors, which try to attach themselves on the active site to inhibit the reaction enzyme. So this is not correct option. Now I'm moving towards the B. It can attach to a site other than the active site. Yes, it will attach itself on the other sites, and then in this way there will be the conformational change in the shape of the enzyme molecule, and due to the change in the shape of the enzyme molecule, there will be the enzymatic reactions can stop. So this is the correct statement. It can attach to the active site and other site simultaneously No, this is not true. This statement C is also not correct because this is not the characteristics of a standard non-competitive inhibitor. That is the it will attach itself on both at the time of active site. So active site uh, it is non-competitive, not attach itself on the active site. Again, I have told you in the statement first, it can attach to either the active site or another site. No, this is not its property that it has to attach itself on the active site instead of the other site. So active site or another site, it don't has any option. It don't has any option. So this is too broad and does not specifically define a non-competitive inhibitor. So non-competitive inhibitor is not of this kind. So only B is the correct option, in which a non-competitive irreversible non-competitive enzyme will always try to attach itself on the uh, other side uh, on the other side. So this binding causes conformational change in the enzyme, conformational change. So it can cause conformational change, and due to conformational change, the it will inhibit the enzyme action. So B is the correct option. Now we are moving towards the next question, and next question is, before mitochondria are extracted from cells for microscopy, they are usually kept in a 0.25 molar decimeter -3 sucrose solution. Why is the sucrose solution used? It is kept in the sucrose solution when they are extracted. So first option is to act as a solvent. Solvent can dissolve the other substance into it. So while sucrose dissolves in water to form a solution, so simply acting as a solvent, it is not a primary reason for choosing sucrose specifically for this concentration for mitochondrial extraction. So many substances could act as a solvent. The key is the osmotic properties of the solution. So this is not the basic reason or the primary reason for this. To enable the rate of reaction of the mitochondria to be determined, to enable. Uh, Again, this is not correct. The primary, you can say, uh, the primary goal of the extraction for microscopy is to preserve the organelles structure. To preserve the organelles. Which organelle? The mitochondria. So So in this case, while mitochondria are involved in reactions, the specific step of using sucrose is about maintaining this integrity. This is not correct. So prevent the mitochondria from changing in dimension. Yes, it can be the correct option. How it can be the correct option? You know, uh, the mitochondria has semi-permeable membranes around itself. So if mitochondria are placed in a solution with a different water potential, what happens when there is different water potential, when there is, if the external solution or the external water potential solution has the higher water potential, so it will enter into the mitochondria. So due to its entrance, its shape will be changed. On the other hand, if the if outer water or outer solution has less water potential, so what will happen? The mitochondria can shrink. So in this way, the shape of the or the dimension of the mitochondria can be changed. So in this way, the 0.25 molecular decimeter solution is isotonic solution. This is the isotonic solution. And the purpose is, and in this solution, which has isotonic potential, so there will be no change or there will be no entrance of water into the or solution into the mitochondria or out of the mitochondria. So there will be no change in the shape. Next is to provide a source of energy, while sucrose is a sugar and could theoretically be metabolized for energy that is not stored during the extraction process for microscopy. So this is not the correct option. Only C is the correct option. Question number 15. Question number 15. The photomicrograph has been shown. Which statement about these cells are correct? So here you can see they have given us the three statements again. So you can see the red blood cells are on screen. So which statements about these cells are correct? So first of all, I would like to move towards the statement number 1. So statement number 1 is oxygen diffuses through the phospholipid bilayer. So phosphored, the function of these red blood cells is the transportation of oxygen. So during gas exchange, the oxygen is a small non-polar molecule. So cell membrane, including of these red blood cells are, they are phospholipid bilayers. So small non-polar molecules can readily diffuse directly through the lipid bilayer down their concentration gradient without the need of protein channels or carriers. So this statement is correct for this phospholipid uh, bilayer of these red blood cells. Sodium ions diffuse through the phospholipid. Sodium ions cannot diffuse. Why they cannot? Because they are larger in size, so membrane permeability will not allow them to pass through. So the phosphovolapile is largely impermeable to these types of ions and other charged or larger polar molecules. So because of its hydrophobic interior, ion required specific protein channels or carriers. So it will not diffuse. So this statement is not correct. The statement number 3 is the water passes in and out of these cells by osmosis. So it passes these by through osmosis. So water, is a polar molecule, but it is small enough to pass through the phospholipid bilayer. So primarily via osmosis, and in osmosis, we always say that the water moves from its higher water potential towards the lower water potential through this phospholipid bilayer. So this statement is also correct. So statement number 1 and statement number 3 are both are correct, and statement number 2 is not correct. According to this, C is the correct answer. A, B, and D are not the correct options. Now moving towards the question number 16, and question number 16 is which row is correct for parts of the phospholipid molecules. So here you can see can be saturated or a phospholipid molecule. And in phospholipid molecules, I would like to discuss its structure first. Phospholipid molecule, it consists of hydrophilic head. Hydrophilic head. Hydrophilic head means water loving. It will be attracted towards the water. So hydrophilic head, and tail. So head and tail. The first row is head and tail. Uh, Phospholipid molecule consists of hydrophilic head and tail. So according to this, I would like to say that uh, can be saturated or unsaturated. So these tray are the tails are three tails are there. So these tails can be saturated or unsaturated. So, the triglycerides are composed of a glycerol backbone and three fatty acid chains. So they do not have a phosphate head group. Therefore, the tail fatty acid chains of a phospholipid can also be found in the triglyceride, but the head cannot. Let's evaluate. For this, how you can see this is not correct. So saturated head will not be saturated. Tails can be saturated, can be found in triglyceride. Yes. And then we move towards the row tail, yes, can also be found in a tail, but head will not. So again, saturated head will not or head will not. So tail can be saturated or unsaturated and can also be found in tails can also be found in triglycerides. So this is the correct option because the tails will be saturated or unsaturated. Saturated in that case when there will be no double bond. When unsaturated means when there will be the double bond. So can also be found in a tri. So the tails will all are always found in triglycerides. So D is the correct option in this case. Now moving towards the next question, number 17. And question number 17 is, a student filled dialysis tubing with the sucrose solution and noted both ends. This formed a cylinder with the length is 50 centimeter, radius of 2 centimeter. So we have to find out the correct answer. So for this we have to calculate length is equal to 5 centimeter and radius is 2 centimeter. So for this, we have to find out the surface area to volume ratio. So surface area to find out the surface area to volume ratio, first of all, we have to calculate the volume. So volume is equal to pi r square h. So volume is equal to pi is equal to 22 by 7 and r is radius is 2. So we will take the square of this 2 and h is equal to 5. So after calculating this, it is nearly equal to 63.62.83. Now we will move towards the surface area. First of all, we will take the lateral surface area. It is equal to 2 pi r h plus 2 pi r square. So first of all, we will calculate 2 pi r h is equal to 2 pi into 5 into 2. So it is equal to 20 pi. And on the other hand, the lateral side said the 2 pi r square. Now we are calculating the 2 pi r square. So it is equal to 2 pi r square is equal to 2 pi into 2 into 2. So it is equal to 8 pi. So the area is equal to 20 pi plus 8 pi. which is equal to 28.5 and it is equal to 87.96. So surface area volume ratio is equal to in this way, that is 87.96 by 62.83. So it will be equal to nearly equal to 1 by 4 is to 1. 1.4 is to 1. So according to this calculation, C is the correct option. C is the correct option here. I am calculating this 87.96. Yes. So it is equal to 1.4 is to 1 ratios. You can write in this way. So this is C is the correct option in question number 17. Now I am moving towards the next question, which is question number 18. And in question number 18 is which process does not involve mitosis? So they have given us the 4 different options. So first of all, what is mitosis? Mitosis is a cell division in which the chromosome number is maintained generation after generation, and one cell is divided into 2 daughter cells. And each daughter cell has the same number of chromosomes and the same type of chromosomes, and the cells which are formed as a result, they will be identical to each other. So asexual reproduction involves the mitosis. And we have to find out which is not involving. So growth of unicellular organisms. Unicellular organisms, grow by increasing their cell size and they're dividing by mitosis fission, which is in prokaryotes to produce more cells, thus increasing the population biomass. So it does not involve the mitosis. So they are not involving the mitosis. So according to this statement is correct. Repair of tissues by cell replacement. So this is essentially the same concept as repair of tissues, in which the you can say damaged tissues or replacement of damaged tissues, and uh, both they involve the same concept as repair of tissues. One body constantly replace old damaged or dead cells through mitotic division, so it does involve mitosis in both cases. So only B is that option which does not involve mitosis. So B is the correct option in this case. Moving towards the question number 19, and question number 19 is the photomicrograph shows cells in the onion 2 tips. Which labeled cells are in anaphase, metaphase, and prophase? So I'm moving towards the X. Here you can see, first of all, what is prophase? Prophase prophase in which nuclear membrane disappears. Chromosomes are randomly present and visible and condensed and visible. And in metaphase, chromosomes are at the equator, in the center, or in the center. And in anaphase, movement towards the opposite poles. So according to this, X is the prophase in which you can say a nuclear membrane is disappearing, or it has, and they are condensed, chromosomes are condensed and randomly present. And Y, you can see it is metaphase as they are present in the center on the equator. And in Z, they are moving towards the opposite pole, so this is the anaphase. So according to this, the answer is anaphase is Z, metaphase is Y, and X is prophase. So A, B, and C options are not correct. Only D is the correct option. After this, question number 20. The diagram shows an outline of the mitotic cell cycle. So mitotic cell cycle, you can see is three different stages, 1, 2, and 3. And after the 2, there is cytokinesis. So which numbered stages of the cell cycle include a period of time when each chromosome consists of sister chromatids joined by the centromere. So here you can see this is a cell cycle with different phases. So interphase, there is interphase, longest phase of this will be interphase, and then G1 phase in which cell grows and perform normal metabolic functions, and S phase in which the DNA replication occurs, and G2 phase is the cell continue to grow and prophase by mitosis, and M phase when mitosis occurs. So the prophase, chromosomes, metaphase, anaphase, all they telophase, all that takes place over here. So here in you can say major phases, there is phase 1. So this is a large segment representing interphase, and phase 2 is the smaller segment represent mitosis. So it will represent the interphase, and 2 is representing the mitosis or M phase, because after this there is cytokinesis. So division will take place. So what happens with this, and phase 3 following phase 2 would also part of the interface. So it can be the part of interface, you can say, but which number? So we have to discuss that first of all, the stage 1, it represents the as I have told you. So what happens during these 3 stages? So 1 and 3 together represent interphase. So interphase, and then you can see the phase 2 is clearly metaphase, mitosis and chromosomes have sister chromatids and prophase and metaphase within M phase. So sister chromatids are present in this mitosis phase, and one is interphase in which the chromosomes, uh, you can say, they are copied and they are attached with with the sister chromatids joined by the centromere. So these phases 1 and 2 are those phases when we can see the chromosomes consist of sister chromatids which are joined by the centromere. But in until anaphase, chromosomes have two sister chromatids till anaphase, before anaphase. So we can say the major A is the correct option. B, C, and D are not the correct options. So according to this, the labeled parts 1 and 2 are the correct options. So according to this, so it was all about from question number 11 to 20. Students, feel free if you want to ask any question regarding these papers or regarding your syllabus. I will always be available. If you message me, then I will reply you in any case. And please don't forget to like, share, and subscribe my channel, and you will be able to get more videos on your subjects. Thanks for watching, and in the next video, I will discuss the question number 21 to question number 30. Thanks.